Now, the extent of the host involvement can help us diagnose the disease. So local infections are limited to small areas of the body, boils, abscesses, etc. And when there's not systemic involvement, we can say that this is like a subdermal parasite, or, if there is more systemic involvement, we can say like a fever plus a pustule. We can say this could be indicative of a virus or a bacterium. So the systemic or the generalized infection is throughout the body. So like measles. Measles, you're going to have fever, body aches. Measles comes from the word miserable, and it's just going to feel absolutely awful. And then you have focal infections. These are systemic infections that began as a local infection. So like, for example, the local abscess could be a result of an abrasion on your skin that was from staph, but it hasn't gone systemic yet, right? So if it's left untreated, however, you'll start to get full body fevers or other systemic associated detrimental side effects as a result of that. So that's a focal infection. So focal infections can start out as a local infection and then progress from there. Sepsis. This is the toxic, and again, these next series, you just have to know the definitions. So create flashcards, drill what these mean. Sepsis being toxic inflammatory conditions arising from the spread of microbes, especially bacteria or their toxins from focal infection. Bacteremia, bacteria in the blood. Septicemia, also known as blood poisoning or growth of bacteria in the blood. You have toxemia, toxins in the blood. Tetanus. A couple others would be Bacillus. Oh no, I'm sorry. Clostridium botulinum. That would lead to toxemia. Viremia just means viruses in the blood. So, for example, not every virus, when you get infected with it, has a viremic stage. When you get influenza pretty much just stays in the lungs. There are a few documented incidences where influenza has gone viremic or the virus has gone into the blood, but it's very rare and not traditional presentation. Primary infection, again, acute infection that causes initial illness. Secondary infection, so this is the infection after the primary. So let's stick with our influenza example. So again, your primary infection with influenza is influenza in your lungs. It does damage to the cilia in your lungs that prevents respiratory mucus from being moved. As a result of that, once the virus is gone, you have pools of warm mucus in your lungs that allow bacteria to come and grow, and you get your secondary pneumonia. In AIDS patients or stage 4 HIV, we get all kinds of different opportunistic infections. You get thrush or yeast infections of the mouth. You get Kaposi's sarcoma, which is a skin cancer that's a result of a herpes virus. You can get pneumocystis pneumonia, which is a fungal pneumonia in the lungs. And then subclinical means there's no noticeable signs or symptoms. Now, the more subclinical, or the higher the probability, the higher the percentage of subclinical infection, the harder a disease is to control. So, for example, chlamydia should have a subclinical or asymptomatic rate of anywhere from 55 to 60%, which means that those people can continue to spread illness without any noticeable signs or symptoms. And that is a big problem. Here in your example here, we have polio and hepatitis A can be carried by people, and those are going to be your spreaders who never have symptoms. One of my favorite examples is noro, the winter vomiting disease. People can go to work totally asymptomatic. About 30% of people who get noro are asymptomatic. Maybe they don't wash their hands when they use the restroom, and they go to prepare, or they inadequately wash their hands. They go to prepare food, and people think it's food poisoning when it's not traditionally food poisoning. in that there are toxins from the bacteria seeping into the food. So, what can make your body more susceptible to a disease? Well, here's the bad news. It varies by disease, and it varies by makeup of the person. So, I've talked about this in class. Women are more likely to clear hepatitis C than men, naturally. So, in this situation, gender is explaining some of the susceptibility. Women do the same thing for West Nile. West Nile virus is almost always more severe or present in men. Women are more likely to clear it, which again, a virus of the same or not same category, of a similar category and similar relatedness to hepatitis C in its genetic structure could explain that. Inherited traits, so you can have sickle cell, predisposing you to other conditions, climate and weather. So if your climate predisposes you to supporting a mosquito like Aedes aegypti, you can have transmission of dengue, yellow fever, chikungunya, Zika, right? Because here in Fresno, we had the right climate, we had the right weather, and then it just took somebody to bring the mosquito here, and now we have Aedes aegypti. Now, fortunately, we don't have enough people with the disease to actually infect our local mosquito populations, but that could change. If you're exhausted, if you've been studying micro and physio too much, you can be more susceptible, and that's from cortisol reducing your immune system. If you are on chemotherapy, and that doesn't just mean cancer meds, if you're asthmatic and you're on inhaled corticosteroids or prednisone, that's doing the same thing as the glucocorticoids in your adrenal gland, which is reducing your immune capability. Age. As you age, your immune system diminishes, and you are more prone to certain types of infection and less prone to others. Like I've said previously, most 70–80% of STDs are in people under the age of 35, 36. So STDs generally drop off. Now, there was a situation in which we've had outbreaks in elderly communities because they thought, they're not at risk, and they were in their twilight years, and they had unprotected sex too much. One person came in with an STD, and it spread like wildfire in that population. And poor nutrition or lifestyle factors can also predispose you. So let's talk about the steps. You have the incubation period. Now, incubation periods vary from disease to disease. Norovirus is like 12 to 24 hours, very quick. Salmonella can also be as little as 12 hours, so it can be very fast. Whereas you can talk about something like hepatitis B, which can be over 100 days. Hepatitis A can be up to 90 days. The prodromal period is the period in which incubation and early mild symptoms occur. Now, the longer the prodromal period, the harder it is to identify what you ate, potentially if it's a foodborne illness, to what gave you the illness. Think about it. Hepatitis A is fecal-oral, sometimes foodborne. If you had 90 days, can you remember what you ate every day for the last 90 days? It can be a little bit challenging. Period of the illness, usually when the disease is most severe. And then period of the decline. Now, in some situations, the period of the decline, as the signs and symptoms are going away, you could still be infectious. So with norovirus, the peak time of infectivity is after symptoms have already gone away. And then you have period of convalescence or convalescing, where the body returns to its pre-diseased state. So this is the general timeline. All of these periods are variable based on the disease that you're talking about. So in the library, you have the Hayman text, the Control of Communicable Diseases text. You can come up to my office and borrow my copy as well. That has a breakdown of these different areas if you want to include it in your paper. So the next topic that we want to talk about is the reservoirs for infection. So continual sources of infection. So humans can act as reservoirs. They can be carriers. They can have inapparent infections or latent infections. Typhoid, you can carry it in your gallbladder and continue to spread it for the rest of your life. There's a story in your book about Typhoid Mary. AIDS, you can have HIV for 10 years and not have any symptoms and continue to spread it. Gonorrhea, you can be asymptomatic, and humans are the primary reservoir. Amoebic dysentery, humans and the environment are the reservoir. So they're free-living organisms that accidentally get in us, and we can act as a reservoir for infection. Animal reservoirs, so zoonoses are transmitted from animals to humans. So rabies and Lyme are two excellent examples of primary zoonoses. And then we have non-living reservoirs, such as soil or water for typhi, cholera, and soil for botulinum. So each one of these can vary. You have good reservoirs and then you have bad reservoirs. A good reservoir means that the disease doesn't kill it. A bad reservoir is the disease kills it. And then they have something called a dead-end host. And so when we're talking about diseases like West Nile virus is a zoonosis, okay? It's primarily in birds. That is the reservoir. But some birds are good reservoirs, okay? So they can have West Nile virus and they do okay and they don't immediately die. And then you have reservoirs like corvids, which are crows and ravens, which die almost immediately from West Nile virus because the infection is so violent. So that's a good and a bad. Now let's say a mosquito picks up that virus and then bites a human. Well, that human is what we call a dead-end reservoir, incapable of reinfecting that animal. I'm sorry, reinfecting a mosquito. Same with horses. If a mosquito bites a horse with West Nile virus, incapable of reinfecting, dead-end reservoir. So these next few slides are a list of organisms, and I'm going to have some matching on the next exam. So it's a good idea to kind of familiarize yourself with the disease, the causative agent, and the reservoir itself. Also, the mode of transmission. So each one of these, as I was mentioning previously, will be matching on the next exam. So you'll have to know either the causative agent, the causative agent, not going to spend a whole lot of time on, but the reservoir, the disease, or the causative agent, and how it's transmitted. We'll talk a little bit about transmission here, but this table 14.2 is very important for you to drill and study. You have 14.2 continued. Again, the different diseases. This will come in handy as we transition to the last unit of the course. Then we have transmission. So how is it transmitted? So please understand that oftentimes if it's transmitted by droplet, it could be transmitted by direct contact. There are a few exceptions. For example, with flu, flu really needs to be inhaled. So as long as the droplet is aerosolized in some way, then is breathed in by somebody, you're not really going to get flu by licking your hands because the virus, the envelope, will get degraded in the tongue. It has to be inhaled somehow going straight to the lungs. Direct contamination or direct contact is, person to person, usually kissing, touching, sexual intercourse, whereas indirect can be by fomites. So norovirus, kind of classic example here. Tissues, bedding, drinking cups, syringes, etc.